Hello everyone, welcome back to another session of Regeneration Biology, and today we will learn a new model that is called Hydra, which has been well explored and well studied in the field of regeneration biology, and it is a quite interesting animal. And humans, before even they start doing research on any subject, say hundreds of years ago, especially with regard to biology, people have observed that the hydra is a very interesting animal and it has the capacity to regenerate. So a lot of mythological connection also is there. Those who follow Hercules, though there is a monster animal that's name also is hydra, but hydra in general it's a small animal, but lot of research interest has gone into hydra in the past years, several years. So let us learn how hydra is a good model in this coming few slides. So as you can see here, regeneration in hydra takes place using two technical methods, or two technical terms are used to describe the regeneration of hydra, that is called morphallaxis and epimorphosis. So we will see more in detail about these two terminology, but I can explain now itself a little bit. Morphallaxis is the ability to form an entire organism from a group of cells. Say you may have heard about plant tissue culture. What the people do plant tissue culture? They take a small piece of the plant tissue and they simply put it in a medium and you end up getting a new plant. Not that a large quantity of tissue, you will end up getting a new plant. Same way, if you take a hydra's tissue, small tissue, and you just put it in a petri dish, you will end up getting a miniature hydra. So this is morphallaxis. But what is epimorphosis? Epimorphosis in the simplistic sense is replacing the damaged portion. If you lost one finger, you don't want the whole animal to come out of that area. If you lost one finger, you want that finger back. So this is called morphallaxis. Okay, we have seen about this different category in one of the earlier classes, but we will revisit this terminology as and when it gets. So hydra is a genus of cnidarians found in freshwater, and most are roughly around half centimeter. That means around 3, 4, 5, around 5 millimeter in its length. And it has got a tubular body, a cylindrical body, and with a fan-like structure, as you can see in this picture, around its head. And there is a head and a distal end at the extreme end of the body, and it consists of a conical hypostome. Hypostome is nothing but its mouth. So this around the mouth, they have a ring of tentacles. So in water, these tentacles will be turning around like a fan and it will create a whirlpool, and the food particles that is floating will directly come into its mouth. So it's a simple procedure what hydra will use for catching its prey, and it's a very miniature animal. And there is also a foot that is located in the bottom. As you can see here, this is the foot region. There is a foot, and it's also known as the basal disc. At the proximal end, it is also known as the basal disc, and it helps the animals to stick to a substratum. It's a rock or a solid wall, etc. Otherwise it will fly off or flew away with the water current. And they have a diploblastic body. Diploblastic body means it has got ectoderm and endoderm. There is no mesoderm. That means normally every organism have got ectoderm and endoderm to start with, and the mesoderm is formed because of an induction event. That is a intermediate tissue that is in the middle of the body that is formed, but they don't have a dedicated mesoderm, but they have a mesoderm like structure, and they are usually called diploblastic. If it has got all the three layers, we call them as triploblastic animal, but hydra is a diploblastic animal having two germ layers, ectoderm and endoderm. Though they do not have a true mesoderm, they have secretory cells, stinging cells, that is nematocytes. That means it can create small amount of toxins to evade unwanted intruders, and gametes and neurons that are not part of neither ectoderm nor endoderm, but they are located in between, and it is often referred to as mesoglia, not mesoderm. And these cells are present, but it's not a true mesoderm. So if you look closely, you can see the Hydra has got a unique phylogenetic position. Like you can see here, starting from Porifera. Protozoa is a single cellular organism, and the next level is Porifera, and then it is classified into multiple pieces. That is, Ctenophora is there, and Bilateria is there, and then there is a Hydrozoa, which the Hydra belong to, and Scyphozoa, Cubozoa, Anthozoa, etc. Anthozoa, normally the jellyfishes all come into that category, and in the group Cnidaria, that is, all this group has got a nematocytes that they have the stinging cells, and the Hydrozoa has the Hydra animal. And here you can see here there is a cartoon of Hydra, which is here. It has got a foot region, and then soon after foot region it has got a budding zone. What it buds? New Hydra can bud by asexual reproduction, and then it has got a gastric region, and then it has got a tentacular region. Right in the center of these tentacles it has got the hypostome. So this is a typical structure, morphology of the Hydra. If you take a cross section, you can see it has got a typical endoderm layer and typical ectoderm layer, and it has got a tiny myofibrils, which is not a dedicated muscles, but it can make some tiny movements, and it is nothing but the mesoglia. And you have dedicated epitheliomuscular cell, they have got sensory cell, and they do have some dedicated septate junctions, and neurons are there, it can sense the environment, and also have got the nematoblasts and interstitial cells and nematocyte, which can produce this toxins, and glycocalyx, and inside also it has got cilia, different secretory gland cell, because whatever food it is eating inside, that has to be digested by these secretions coming from the endoderm cells. So it is a very interesting, unique animal, but it has got almost all cell types that is required, and because this is very important for us to understand when it comes to morphallaxis and epimorphic regeneration. If you look closely, you can see the Hydra can reproduce sexually, but they do not usually do sexual reproduction. They always does the asexual reproduction, that is called budding off. As you can see in this picture, this is a single Hydra, and a small miniature Hydra is coming from the surrounding. This is called budding. Hydra's body is not stable. As you can see in this picture, you see 8 days, 2 days, 20 days, 4 days is written. You may wonder what does it mean. It simply means the cells of the body are constantly dividing and are displaced to the extremities of the column from which they are shed. So extreme tip, the new cell is dumped off, and the new cell keep proliferating and keep replacing. Thus the Hydra's body is always regenerating. You can tell in a realistic manner, you can say Hydra's body is always regenerating. Like our body also, our skeleton has got a lifespan of around 7 years. If you are 7 years old, you are on the first version of your under 7 years, first version of skeleton. If you are 70 years old, you are on the 10th version of your skeleton. So your whole skeleton is revamped throughout the year, but Hydra can produce every cell. So as you can see here, this is the hypostome area. 20 days it takes to move a cell. That means the movement is limited. And this tentacle region, it takes 4 days from here to reach the extreme tip. It takes 4 days. And in this area around the neck region, little or no cell movement, because that is a place where the cells are confused. Should I go upward? Should I go downward? And here somewhere in the middle region, somewhat the say chest region, if you compare to that of a human, around 8 days it reaches to reach all the way up to a budding region. Here it takes around 2 days to reach all the way up here. And from this lower mid region till the foot, it takes 20 days. That means the rate, although the distance is short, the movement is low. So this is on an average, because you need to know this is the movement of cell. That cells are being formed here, and the cell formed here now takes 8 days to reach up to here. And cells that is formed here, it takes around 4 days, and then it fall off from the tip. And here it is movement is less, hence it is taking 20 days around the neck region. But Hydra's body is always regenerating. So this cellular replacement happens from 3 cell types: the ectodermal, the endodermal, and they constantly divide to produce more of the epithelial cells. The third cell type is a multipotent interstitial stem cell that is found in the ectodermal layer, which generates the gametes, neurons and secretory cells. So there is a dedicated stem cell which is helpful in the constant replication, and it is called interstitial stem cell. So the three cell types are enough to form new hydra. That means you need to have an ectoderm, endoderm and this interstitial cell. If you put them together, the new hydra will be formed. Why? Because interstitial cell is good enough to give rise to gamete producing cell, neurons and other secretory cells. So this way the hydra, if you disassemble into a mass of tissue, say 50 cells or 100 cells of hydra, it can give rise to a miniature hydra which will eventually grow bigger. So if you look closely, the regeneration capacities of hydra is a Hydra vulgaris, which is a species. The ability of hydra to regenerate when dissected in various manners and various morphological changes observed are shown here. See, this is a hydra as you can see here, and one scissors picture is given. That means normally in food packet you will see a scissors symbol. That means that is the place where you should cut it. Same way, if you cut it through this plane, left half and right half. So what the left half did, it produced the right half. What this right half did, it produced the left half. But important thing to note is, it simply do not produce missing part. Eventually it produces the missing part, but with the availability of this entire left half, it made a miniature whole animal. Say for example, let us assume the hydra is 1 kilo, easy. So you cut it into two pieces, 500 gram and 500 gram. Now this 500 gram hydra made a new hydra which is exactly 500 gram with a miniature hydra. Now it will try to reach the size of 1 kilo. So this is what you are seeing. Say this central hydra is big and cut into left half and right half, and this newly formed hydra is exactly half the size of this parent hydra. That is longitudinal division. Now you see transversion division. Head one piece, belly one piece, foot one piece, cross cutting. You got, let us assume this 1 kilo hydra you made 330 gram. And now you end up getting a tiny hydra. Head region made a full hydra, which is a miniature hydra. Body region made a hydra, which is again a miniature hydra. The foot region made a hydra, which is another miniature hydra. So the newly formed three hydra, they are identical. They look like clones. But not that it is smaller than this hydra, which is made by two cut. That means 500 gram hydra is bigger than the 300 gram hydra. So this is technically we refer to as morphallaxis, and ideally in effect it is epimorphosis, because the left half was missing, the right half formed, but it's not identical size, but eventually it will become that size of that of the parent. So hydra can regenerate missing parts upon transverse and longitudinal dissection, or you can do whichever angle you want to cut it, you can cut it. The hydra give rise to a proper full-fledged hydra. Experimental embryology, or one might say the experimental biology of hydra, began with Abraham Tremblay's work on hydra regeneration, that is done in 1741. That means almost 300 years ago. People knew about the hydra regeneration in a recorded form. Maybe people would have noticed much before that. If hydra is cut into as many as 40 pieces, they are reborn as many complete animals similar to the first. Similar, not size wise, appearance, morphology wise. Each piece would regenerate a head at its original apical end and foot at its original basal end. They will be miniature hydras. Every portion of hydra's body along the apical basal axis has the potential to form head and foot. Every piece has the potential to form, whether it's a belly piece, neck piece, body piece, foot piece or head piece, doesn't matter. However, the polarity of the organism is coordinated by a series of morphogenetic gradients. Gradients means they are present in different concentration. Say if I say head gradient, if I say in head it is maximum As it move away from the head, it is going to be lower, 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 and towards the foot region, it is the least. So that is same way there can be a food gradient. In the food region, a given chemical, given molecule will be highest in concentration, and it becomes lower, 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 and lower. This gives a positional identity. So whenever you take a slice, you will find a place where both the gradients are equal. You will see one gradient is 50, another is only 30, or another place you will see one gradient is 80 and the other gradient is only 20. You will find based on the gradient of different molecules. It's not just two molecules, there can be multiple molecules, and same way it can have left to right gradient, right to left gradient, back to front gradient, front to back gradient also is possible. They are called morphogenetic gradients that permit the head to form only from one place. When you take a body piece of Hydra, you won't get three heads all over the body. One head in the above actual place, one on the left side, one on the right side. You don't get like that. You get only one head, no matter what is the slice size. This is something very interesting. One place of the basal disc, and it's also important to note that the head form only one. In the head region, it will not produce, same tip region, say anterior region, it won't produce two heads. It will produce only one head. Like you can see here, some experiments are done to demonstrate that regeneration capacities of Hydra vulgaris. Hydra vulgaris is a species that is used to study the regeneration capability. Like this is a full Hydra, and this you macerated, like you made into, you made a squash out of this Hydra, made into cell suspension. And now, soon after, these cells are kept in a liquid medium. Medium means simple water. They will assemble to form a circular shape. That means it forms like a football. It formed a sphere. And now this sphere is slowly changing its shape to that of a typical Hydra. And if you see in this cartoon, what you are seeing here, you can make a cut anywhere. Say you cut it near the neck region in this axis. You are left with here. Immediately it heals. And somewhat like that of a blastema structure is formed within around half an hour to 3 hours. And around 3 to 8 hours, the continuity is established. Like here, there is no continuity. Here, the continuity of mesoglia or mesoderm is established. And by around 30 to 36 hours, it will start developing the tentacles slowly. And later, it gives rise to a head, proper head with a hypostome that is being formed. And this is the normal method. Roughly around 1.5 days, the new Hydra, miniature Hydra is formed from a mass of cells. So regeneration from dissociated and reaggregated cells are shown in the top panel, and this is bottom panel is the kinetics of growth morphological changes during Hydra's head regeneration. Now you can see there are some evidence about to show the importance of the gradient. We said that there is morphogenetic gradient. Can we prove it? For such gradients was obtained from the grafting experiments done by Ethel Brown in early 1900s. That means more than 100 years ago these grafting experiments are done. Like you can see here, this is the head region, like hypostome. You take a portion of hypostome and you attach onto the trunk region of another Hydra. So the donor hypostome is taken and attached onto the trunk region. That is, you are sticking something onto your belly, and you see a new head is being formed from that region. So the hypostome only you put in. There is no injury, mind it. You just transplanted the hypostome region onto the belly of another recipient. When the hypostome tissue from one Hydra is transplanted to the middle of another Hydra, this transplanted tissue forms a new apicobasal axis. Because you put a hypostome has got a strong concentration of the head gradient, and that is dissolving into the belly region, and you created a apicobasal axis, and with the hypostome extending outwards, and that will establish by movement of the hypostome away from this body. So how it will move? It cannot fly. It will slowly keep synthesizing cells and keep walking out of the body, and you end up getting a new Hydra, just like a budding, as you saw in this case. Same way, let us take a basal disc. Took another donor Hydra, took the foot and attached it into the same belly region. And you see, what do you see? Now, instead of a head coming out, you see a foot that is coming out. That means there is a hypostome. There is a basal disc has got another gradient. Like hypostome had one head to tail or head to feet gradient. Feet to head also there is another gradient. When a basal disc is transplanted to the middle of another hydra, new apico-basal axis forms, but with the opposite polarity. With the basal disc is now extending. Head is not coming because you did not put hypostome, you rather put a base or a feet, and now the feet started moving out. In both head and both feet, it moves away from the donor's body while attaching, holding on to its body. That means the strong gradient is trying to establish the actual gradient by moving away from that site. And you can see here the new feet is formed from the belly. Now, you can see, now if you take from a donor, you took a head and half base together, you put both together onto a donor in the same place. That means if you are taking a portion of hypostome and you are taking a portion of basal disc, both are put it in one implantation, together you put it. Then what you end up getting, you will get either a weak apical induction, not strong, it doesn't grow very strongly, a weak apical induction, or a weak basal induction, or if it is absolutely, why you got a weak apical induction? Because although you took 50-50, maybe little bit by one cell or two cell was little more in the hypostome, hence it get a weak head induction. And why you get a weak basal disc induction? Because maybe basal cell was little high. But if it is absolutely precise number, 100 hypostome cell, 100 basal disc cell precisely, then you get zero induction. There is nothing coming out of that body. That means it got neutralized, it got nullified. So there is called a no induction. So when tissues from both the ends are transplanted simultaneously into the middle of another hydra, either no new axis forms or the new axis has got less polarity. That means some cancelling out is happening. That means one gradient want to take over, the other gradient is suppressing it. That is why you get weak or nil. These experiments have been interpreted as follows. There is a head activation gradient, that means favoring the head, highest at the hypostome. Head activation gradient is highest at the hypostome. And there is a foot activation gradient that is highest at the basal disc. This gradient can be there anywhere in the body, but the highest of head activation near the hypostome, highest foot activation in the basal disc. That is the inference you can take from this. The head activation gradient can be measured by implanting tissue rings from various levels of donor hydra into particular region, wherever you want to, of the host trunk. The higher the level of the head activator in the donor tissue, the greater the percentage of implants that will induce the formation of new heads. Instead of going into the proper hypostome region, or instead of going from the proper foot region, you can go closer around the neck region. Say, considering human, so your head, and your neck is closer to the head. Chest is little bit more closer to the head. Belly is somewhere in the midway, right? Same way, you take a tissue disc from closer to the head and you implant anywhere into a donor hydra, then there is a higher chance it will form a head, not the foot. Same way, if you go to the lower half, closer to the foot region, away from the head, there is a higher chance the foot will come. Not that you need to take an donor tissue precisely from the hypostome or precisely from the basal disc. Even closer is good enough. That means there is a gradient that is into action. That is what the inference. Now, if you see the formation of secondary axis following the transplantation of head regions into the trunk region. We saw different transplantation if you are doing. Can we create a secondary axis? One axis we know. Can we make a like a parallel government or parallel economy, something like that? So many such research are done in embryology and regeneration biology studies. So these experiments are done. One experiment, what they did, hypostome tissue grafted into the trunk induces the host on trunk tissue to become tentacles and head. Now, what do you understand from here? You are tweaking the system. So this is a reference. You can see hypostome tissue is now grafted into the trunk of of the, it can be same animal or it can be another animal of your liking. It induces the host on trunk tissue to becomes tentacles. That means new cells are recruited. That means the hypostome or this gradient had the potential to influence the neighborhood. Just like if I put a fragrant material in a room, it can be a normal room, dirty room, or already fragrant room. Wherever I put it, it influences the neighborhood molecule. Sometimes it can mix, but in this gradient, what happens? The gradient is powerful enough to influence the neighborhood. Like human company, if you are with good company, you can get influenced by that good company, or if you are with the bad company, you can get influenced with the bad company. Same way, the trunk region, which is weaker both for head gradient and tail gradient, because it is somewhat like in a midway. That time you suddenly put a head gradient, then all the cells will start responding to it and they start making head, hypostome, tentacles, etc. So subhypostomal donor tissue. Hypostome is one tissue, subhypostome, you went a little bit below, grafted onto a trunk, cell differentiates into head and upper trunk. So what does it tell you? Below the hypostome also, it has got gradient that is favoring the formation of a head. So what you can see here, these experiments are done by Ethel Brown. He noted that the hypostome acted like an organizer or the director of a film. Organizer means something which can influence the neighborhood. That is the organizer, a leader. Just like how several freedom fighters influence the mass people, like a leader, a global leader influence the public, something like that. That's called organizer, the Hydra. And Brown and Body in 2002 confirmed this by demonstrating that when transplanted, the hypostome can induce host tissues to form a secondary body axis. Primary body axis already exists, it creates a secondary body axis, and that secondary body axis is formed by a small tissue. That is what is more important. And the hypostome produces both head activation and inhibition signal. This is maybe sound a little conundrum, we'll come to that, because when you put a new tissue, it is helping the formation of one head. But you also know that who counts? Why can't form one more head if it is favoring? You can have 5 heads, no? But it doesn't. So it allows, once one head started forming, it do not allow one more head to start forming. So this is something very interesting you should note. The same head favoring signal allows head to form, but it do not allow heads to form. Multiple heads don't form. So the hypostome produces both head activation and inhibition signals. The hypostome is the only self-differentiating region of the Hydra. It can self-establish or self-influence the neighborhood cells, and self-differentiating region, just hypostome region is good enough. It contains a head inhibiting signal which suppresses the formation of new organizing center. So head activation is one thing, head inhibition is one thing. So later people figured out that what are these molecules, etc. But these experiments told the researchers that the signal can be activating and signal must be inhibiting also, so that only one structure will be formed. The scientist Body also found that even transient contact with the hypostome region was sufficient. You take a hypostome, no need to transplant. Take a hypostome from a donor, go and touch into the belly of another donor. Just to touch it and come. No need to keep it there. Was sufficient to induce new axis from the host Hydra. How interesting it is. If you think about the regeneration, how exciting and interesting it is. You are not transplanting, you are not doing anything, just physical touching. Take a tissue, go and touch it. Just like smelling. If you want to smell a fragrance, you don't need to pour fragrant material into your nose. Even agarbatti, if you stick it, if you go pass through, you get the sampling. So this system is so powerful that one time touching, it is done for the rest of the life. So this is why this regeneration biology becomes so exciting and interesting. And we will study more about Hydra regeneration and its mechanism in the next class. Thank you.